Hi, I'm Rachel Silvio, and this signal transduction presentation is about G protein coupled receptor signaling for Chem 530. The objectives are that students will be able to define the parts of the cell signaling pathway in general, explain the structure and function of G protein coupled receptors, apply knowledge of G protein coupled receptors in real-life example of an adrenergic pathway, and use knowledge of GPCR signaling to identify new research for therapeutic applications. A signal transduction pathway is the process where a signal on the cell's surface creates a cellular response through a series of steps, starting with a receptor. Now, we have learned that different cell types can respond the same to different signals or respond differently to even the same signal. Cell communication between cells is extremely important for the well-being and homeostasis of a multicellular organism, but occurs for all types of cells, from unicellular prokaryotic E. coli cells to neurons in multicellular organisms. Evolutionary biologists agree that because this process is so similar in each of these very different cell types, that signal transduction evolved before the development of multicellular organisms did. The function of cell signaling is to alter the behavior or nature of the cell, such as deactivation or activation of a protein. Phosphorylation, which is the addition of a phosphate group, and dephosphorylation, the removal of a phosphate group, are important pieces needed for controlling protein function. The three steps of the signal transduction pathway are reception, involving the binding of a ligand and receptor, transduction, and response. During reception, the external signal, called a ligand, shown here in yellow, reversibly binds to a receptor protein shown in purple. This protein can either be found on the cell's surface, like in this picture, or inside the cell. During transduction, phosphorylation or shape change can activate steps, causing a cascade involving more molecules down the pathway. And the final step of the pathway ends in a cellular response, which is a specific action taken by the cell. Examples of response could be activity by enzymes or the activation of certain genes. Now, the binding of the ligand and protein is specific in size, shape, large, and hydrophobic versus hydrophilic character in all organisms, and cell types in multicellular organisms, like enzyme substrate interactions. The ligand is the primary messenger, and it binds to the receptor. If it is hydrophilic, meaning loves water, the messenger will bind to a receptor in the cell membrane. Membrane receptors will respond to these signals that are water soluble, since they need help to pass through the lipid bilayer of the membrane. If the primary messenger is hydrophobic, meaning fears water, then the messengers will bind to intracellular receptors. The four types of membrane receptors used in signal transduction pathways are G protein coupled receptors, which we will go into more depth in this presentation, receptor tyrosine kinases, RTKs, ligand gated ion channel receptors, and nuclear hormone receptors. Transduction is the next step in signaling after reception. Transduction involves the amplification of the initial signal using proteins, ions, and phospholipids in multiple steps through the intracellular pathway. These pathways have a phosphorylation cascade, with proteins such as kinases acting as relay molecules, as shown in this picture on the slide. The phosphorylation of one protein leads to shape change or phosphorylation of another, and then activation of the next protein in the cascade. And once all the proteins are activated, the anticipated response from the cell is triggered. Now, about 2% of our DNA encodes for making protein kinases. Along with kinases, the protein phosphatase is another important protein in these transduction cascades. Kinases are enzymes that function in phosphorylation by attaching a phosphate group to a protein and activating that protein. And phosphatases are enzymes that function in dephosphorylation by removing a phosphate group from a protein. The inactivation of the protein turns the signal off. So these signals are reversible so that the pathway can reset in order to prepare for another signal and cellular response. The phosphorylation and dephosphorylation cycle allows the cell to have a switch, like a light switch, turning necessary activities on and off as many times as the cell needs. Other key players in signal transduction pathways are second messengers, which we will discuss later in the presentation. The last part of the signal transduction pathway is the cellular response, which can occur in the cytoplasm or the nucleus. In the nucleus, an example of a response would be turning specific genes on or off in order to regulate what protein the cell needs to make through protein synthesis. Now, if this regulation occurs, the final protein activated in the cascade might be a transcription factor, a protein that turns genes on and off by regulating what segment of DNA is used to make the mRNA, or possibly a translational regulator that controls the mRNA translation into amino acid sequences. Another possible cellular response is regulating protein activity, allowing for the cell to regulate both the amounts and the activities of these proteins. Some examples of cellular responses regarding protein activity are controlling metabolic pathways, like with epinephrine that we will be discussing later in the presentation, opening and closing membrane ion channels, and possibly increasing or decreasing catalytic activity. The response could also be unrelated to proteins, such as changing the shape of a yeast cell in order to mate, regulating cell growth, cell migration, or the induction of apoptosis, which is programmed cell death. Now, the extracellular signal, or ligand, is the first messenger in the signal transduction pathway. It can be a hormone, such as glucagon, a growth factor, such as EGF, which is epidermal growth factor, or a neurotransmitter, such as serotonin, that binds to the cell membrane receptor for activation. Now, later down in the pathway, another molecule is activated and acts as the second messenger. This molecule can be a low molecular weight metabolite or inorganic ion, functions in activating or inhibiting additional targets downstream in the signal transduction pathway. Now, these targets are often protein kinases. Some examples of second messengers are calcium, which are calcium 2+ ions that are often stored in the endoplasmic reticulum compartments to use when needed. Cyclic adenosine monophosphate, which is cyclic AMP, a derivative of ATP. There's cyclic guanosine monophosphate, which is cyclic GMP, and also inositol triphosphate, or IP3, that is made from phospholipids. A G protein coupled receptor, or GPCR, is an integral protein with 7 helical domains, or heptahelical, that go through the cell membrane with heterogeneous amino and carboxyl termini. The bottom of the ligand binding pocket on the signal molecule links to the G protein coupling region on the extracellular side of the GPCR. The GPCRs work with an intracellular G protein, named because it binds with the guanosine triphosphate, or GTP. The G protein rotates between the active form when bound to GTP and the inactive form when bound to GDP, during signal transduction. The binding of the ligand on the extracellular side of the GPCR activates the receptor, changing its shape. The GPCR interacts with the nearby G protein, activating it and causing the GTP to replace the GDP. Now, this phosphorylation causes the alpha subunit to separate from the beta gamma dimer of the G protein. And the G protein will bind to an effector enzyme or possibly a nearby membrane ion channel, activating that protein and sequentially activating downstream proteins and ultimately triggering a cellular response. An activated pathway is often short-lived, though, since the G protein is also a GTPase enzyme, hydrolyzing the GTP back to GDP, turning that signal off. The pieces involved in this signal transduction pathway can be reused for another cellular response. An interesting fact, there are about 800 GPCRs identified. About 350 of those are endoreceptors, such as hormone and growth factor receptors. About 500 of them are olfactory, which is smell, and gustatory, which is taste receptors. The top picture here shows the olfactory cells, and the bottom picture shows the gustatory cells that have GPCRs in their membranes. Now, when referring to epinephrine or norepinephrine, we use the term adrenergic. There are 5 adrenergic receptors: alpha 1, alpha 2, beta 1, beta 2, and beta 3. Most sympathetic target cells, such as the cells in the vascular smooth muscles, have alpha 1 receptors for epinephrine and norepinephrine. Cellular response in these cells normally results in increased calcium levels and thus increases muscle contractions. Now, alpha 2 is for the pancreas, platelets, gastrointestinal tract, and salivary glands. Beta 1 is for the heart and kidneys. Beta 2 for smooth muscle, vascular tissue, skeletal muscles, and liver. And beta 3 is for adipose or fat tissue. Now, beta receptors generally alter metabolism, such as breaking down glycogen and fat, which I'm going to use as an example. So we're going to look at adrenergic pathway, a great example of the GPCR signaling. Now, when activated by epinephrine, the beta adrenergic receptor, which is the GPCR here, will change shape, often with large outward movements of the TM5 and 6 cytoplasmic tails, affecting its interaction with the associated G protein. The G protein is called heterotrimeric because it is made of 3 subunits: alpha, beta, and gamma. Now, 2 of these subunits, the alpha and gamma, are attached to the cell membrane intracellularly by lipid anchors. GPCRs act as guanosine nucleotide exchange factors that will assist in switching the GDP to GTP on the G alpha of the inactive trimeric G protein. This causes the signal to be transmitted from the activated G protein to an effector protein, in this case, adenylyl cyclase. Now, because the G alpha protein acts like a switch, it is also called a stimulatory G protein, or GS. The GDP that dissociated from the G alpha protein is replaced with the GTP, causing the G alpha to leave the beta gamma dimer from the trimeric G protein complex and go to the adenylyl cyclase, causing its activation. So adenylyl cyclase is another membrane protein, but the active site on this is on the inside of the cell in the cytoplasm. Now, this now causes adenylyl cyclase to make a second messenger cyclic adenomyophosphate, which is a CAMP, from ATP. The bound G alpha converts the GTP to GDP becoming deactivated, leaving the adenylyl cyclase, causing the cyclase to be inactive once again. So the now inactive G alpha joins with the other two parts of the G protein, the beta and gamma dimer, becoming usable once again, once another ligand binds, and the process can start all over again. What happens to the rest of the cellular response? So in this case with epinephrine, the CAMP activates CAMP-dependent protein kinase. It's also called protein kinase A, or PKA for short. This activation causes the phosphorylation of either a serine or a threonine residue of certain proteins such as glycogen phosphorylase B kinase, or GP. This activated kinase prepares stored glycogen in muscles and liver to break it down for energy in the form of glucose. Now, a second enzyme, glycogen synthase, which is GS, is also involved. This enzyme helps build glycogen. So phosphorylation of this enzyme actually inhibits its activity, preventing glycogen molecules to be made. There are three ways of ending the response. If there is low epinephrine in the blood, it will dissociate from the GPCR, becoming inactive, signaling pathway will stop. The second way of terminating the response is due to the GTPase activity of the G protein. So the return of the GTP back to GDP, which causes the return of the G alpha subunit, and the adenylyl cyclase will no longer be activated. The third way of stopping the response is when the CAMP, the second messenger, is converted to 5' AMP by cyclic nucleotide phosphodiesterase, becoming inactive. But if the epinephrine continues, the signal can be desensitized by a beta adrenergic receptor-specific protein kinase and beta-arrestin. So beta-arrestin will then block the site that the G protein normally interacts with GPCR. And GPCR will move into vesicles inside the cell. So this picture on the bottom right of the slide shows the beta-arrestin in orange, which attaches to the GPCR, causing the GPCR and the surrounding membrane to become a vesicle with the help of clathrin and other proteins. So at this point, the epinephrine can no longer reach the receptor to go through that signaling pathway. As of 2017, 134 GPCRs were targets for approved drugs in the United States. Now, there are around 700 approved drugs, which ends up being about 34-35% of all approved drugs that target these pathways. GPCRs are the largest family of proteins that have drugs that target them, most of which are small molecules and peptides. Now, in order to design drugs that target these pathways, information regarding their structure and mechanism of action are important. Now, there are around 360 endo-GPCRs, ones that are not olfactory, taste, or visual receptors. And of these endo-GPCRs, about 100 of them are called orphan receptors, and those that do not have a known agonist that activates it. So, with all this unknown research, as the research continues, there will be an increase of these GPCR targeted drugs. GPCRs are ideal for therapeutic applications for several reasons. Since GPCRs bind to extracellular signal molecules, there's an increased therapeutic potential of changing the internal state of the target cell without needing that molecule to be permeable through the membrane. Also, GPCRs influence so many functions within the body, from smell to vasoconstriction, that there are so many applications of these therapies. Now, GPCRs have a role in the following human conditions. I want to start with asthma and allergies. Now, G protein signaling works in the immune, nervous, and muscular systems that can cause allergic responses and asthma. So researchers are studying asthma therapies that use GPCR agonists or antagonists. So, one example of an agonist would be the beta 2 adrenergic receptor in order to increase smooth muscle relaxation. And an example of an antagonist would be the cystenyl leukotriene receptor that inhibits nasal passage inflammation. For depression, there are antidepressant therapies. Doctors are focusing on two GPCRs found in the brain. The first one is CRH1, which is corticotropin releasing hormone 1. And the second is a vasopressin receptor called V1B. And these increase levels of the corticotropin releasing hormone, the CRH, and vasopressin, AVP, which then in turn increases glucocorticoids that have been shown to increase depressive effects. So antagonists at the CRH1 and V1B receptors are being developed as antidepressants. I want to talk about blindness. Cone opsins are G protein coupled receptors. So research has been conducted on mice showing that expression of light sensitive GPCRs, such as vertebrate rhodopsin, found in retinal neurons can increase sensitivity, allowing for therapies to help blind mice. So the picture on the slideshow shows the signaling pathway of the GPCR rhodopsin in a rod bipolar cell. Now, there are other human conditions that GPCR has a role in, such as diabetes, cardiovascular defects, and cancer. Abnormalities in GPCR signaling pathways, especially with the protein kinases, are responsible for many diseases associated with the cell cycle, leading to cancer. So in review, first we learned about the steps of signal transduction. Those three steps: reception, transduction, and response. Then we looked at the GPCR structure and its 7 transmembrane helices. We discussed an overview of the GPCR signaling, where an external ligand will bind to the GPCR, switching the GDP to GTP. The G alpha subunit dissociates, which activates or inhibits an effector protein, resulting in second messenger effects and ultimately resulting in a cellular response. We also looked more in depth at an adrenergic receptor example, where it activates a G protein, activating the adenylyl cyclase, increasing the second messenger CAMP, stimulating pKa, that kinase in the enzyme cascade, phosphorylating target enzymes, such as those that will get glycogen out of storage. We also discussed several ways that this signal is terminated. And lastly, we looked at how researchers use drugs to target GPCR pathways in therapies. Here's a picture of the human beta 2 adrenergic receptor. The blue on the right is the N-terminus end, and the red is the C-terminus. Thank you for watching my presentation. Feel free to ask questions or leave feedback.